The overall goal of this procedure is to assess the myogenic potential of induced pluripotent stem cell-derived muscle progenitors in mouse models of acute and chronic muscle regeneration. This is accomplished by first assessing the myogenic potential of the cells in vitro. The second step is to transplant the cells via intramuscular injection. Alternatively, cells are injected in the femoral artery by a microsurgical procedure. The final step is to evaluate the functional amelioration of the dystrophic phenotype of the treated animals. Ultimately, immunofluorescence and histology assays are used to show engraftment and functional contribution of the donor cells to the host muscles.
This method can help answer key questions in the field of skeletal muscle regeneration, such as investigation of cell therapy approaches.
The implications of this technique extend towards therapy of muscular dystrophies, as it could pave the way to autologous cell therapy strategies using iPS cells-derived progenitors.
Though this method can provide insight into stem cell-based therapies, it can be applied to other systems, such as delivery of molecules intramuscularly or intra-arterially, and subsequent analysis of their effect on muscle function and morphology.
We first had the idea for this method when we realized that iPS cell culture could provide a method to overcome some of the limitations of adult stem cells, such as the availability from tissue biopsies and their expansion potential.
Begin by generating a stable cell line of IDEMs that are transduced with a tamoxifen-inducible vector that overexpresses the myogenesis regulator MyoD. After